All right, this is chapter 6, alcohol, our first drug that we'll actually discuss. So one of the first things that we'll do in this chapter is to remind you, if not inform you, but I think you're probably already aware that there are different types of alcohol. Okay, I'm trying to get my screen to advance here, if I can get it to, okay, there we go. All right, so our first type of alcohol is isopropyl. Okay, isopropyl is a rubbing alcohol, so you've probably at some point encountered it, used it, if you've had sore muscles, muscle sprain, so forth. This is our rubbing alcohol. The next type of alcohol is methanol. This is a wood alcohol. Methanol is often found in industrial cleaners. It's sometimes found in things like toners. It's something that's very, very dangerous if humans consume it. And unfortunately, there are examples of, folks who know that this is a type of alcohol, and when they can't get their hands on drinking alcohol, they have, there are examples of individuals that have consumed methanol. Problem is that we can't metabolize that. And so methanol could easily kill you. And one of the ways that you counteract ingestion of methanol is by administering a large dose of ethanol. Okay, the ethanol will act as an antagonist, and it will displace or bump that methanol out of the receptor binding sites. Okay, and so of course ethanol or ethyl alcohol is a type of alcohol that we drink. Okay, and so our drinking alcohol is made by a process called fermentation, excuse me. So with fermentation, you start with a sugary substance, and you have yeast, okay, that's added to that sugary substance, and then of course you have air that's part of the mixture. The metabolic processes of the yeast will convert the sugars into alcohol, and they'll also produce carbon dioxide. Okay, so of course when you're talking about wineries and breweries and so forth, and you've got some sugary substance, carbohydrate base, yeast added to these substances, air in the mixture, and you let the yeast work their magic. Out in nature, you generally would have fruit, apples, pears, grapes, what have you, that may be rotting or so forth, but they're out there in the open air. Wild yeast invade these, work their magic, convert those sugars into alcohol, and of course producing carbon dioxide as well. So there are some stories that suggest that some of the first examples of the behavioral effects of alcohol were humans watching the behavior or having the opportunity to observe the behavior of animals that had eaten rotten fruit. So they saw the animals eat the rotten fruit and then observed the behavioral changes. Now it's a bit curious that a human would say, I want to feel like that animal felt after it ate that rotting fruit, but there are stories that suggest that some of the curiosity arose in that manner. So, just little interesting factoids about alcohol. Okay, distillation. This is a process where alcohol, okay, generally alcohol that may have been produced by fermentation, that is heated, okay, because there is a temperature point where alcohol will vaporize. So you heat up the alcohol to very, very high temperatures, and then the alcohol vaporizes. Those vapors are collected in coils, and then that's cooled, okay? And so what happens when you heat the alcohol to very, very high temperatures, the alcohol vaporizes, the other impurities don't. And so you're separating the alcohol from the other stuff that may be in the liquid. And so the end result is a substance that has a higher alcohol content because you've separated out the impurities. You heat that alcohol to high temperatures. The alcohol molecules vaporize. The other stuff does not vaporize. That those vapors are collected in coils and then they're cooled. Okay, and you know that once those vapors cooled, it's back into a liquid form, and so you end up with a substance that has a higher alcohol content. The examples that you see here on the slide are just there simply to give you an idea of some of the alcoholic beverages that are the result of the distillation process. You will not be quizzed on how brandy's made or how whiskey's made, and so you'll see here for brandy, it's distilled wine. So you would have a wine that's already been made by process of fermentation, heat that up to high levels, and so you're actually able to separate out the alcohol vapors from the other impurities and end up with something that has a higher alcohol content. Okay, and then your rum, distilled molasses. So remember, we start with the sugary substances. Okay. And then you've got fortified wines. These are wines that have extra alcohol added to them. Your sherries, port, madeiras, muscatels. And if you are of drinking age and have had one of those, then you'll notice that people are usually drinking very small amounts of those. Sometimes those are after dinner drinks or sometimes what we call aperitifs. And those tend to have a higher alcohol content than just a regular wine that we might have with dinner. Okay, so let's talk a little bit about alcohol content. Okay, in terms of how much alcohol is in a beverage that an individual may be consuming. So it's usually expressed as percentage figures, either by volume or by weight. So if you have the opportunity to look at the side of a bottle of wine, a wine cooler, if you look at the side of a beer can, if you look at a bottle of liquor, then sometimes you're going to see percent volume, or you're going to see percent weight volume. Okay, now why do we have these two things? These two things could yield differences in the alcohol content of a solution. Okay, now why is that? A molecule of alcohol weighs less than a molecule of water. So if we're talking about a 10% volume solution, that would have less alcohol content than something that was 10% weight per volume. Okay, because if we're talking about 10% of the total weight being made up of alcohol, then that 10% of weight has already taken into account the difference in the weight of a molecule of alcohol and a molecule of water. Whereas if we're just talking about 10% volume, the liquid composition, that value does not take into account the differences in the specific densities of alcohol versus a water molecule. Okay, so when you get the opportunity, just look at the side of some of these. So you may see 4% volume on the side of a beer can, or 5% volume. Okay, but they're typically going to be represented as volume or weight volume, or what you will see sometimes on liquors, or what we call hard liquor. Sometimes you'll see proof on those. Here in America, you're likely to see something that may say 150 proof. That is approximately a 75% alcohol solution. Okay, if you see something that says 200 proof, that's roughly 100%. If you're in Great Britain or some other countries, then you're not going to see 150 or 200. You would simply see 100 proof. 100 proof, roughly, 100% alcohol. If it's 50 proof, then it's roughly 50%. And remember, here in the US, it's roughly half. So if you see something in the US that says 100 proof, it's about 50% alcohol. Okay. And you'll see in your textbook that there are some suggestions that, we convert to a standard international system. I'm not sure how close we are to moving towards that, but I don't think the average person wants to walk around talking about nanomolar solutions. Okay. Almost forgot to tell you some interesting folklore. It may or may not be true about this term proof as well. Legend has it that when alcohol was being sold, one of the ways that individuals could determine whether or not what they were purchasing was really alcohol and had a sufficient alcohol content was that they would take some of the solution and they would wet gunpowder. Okay, and if you wet the gunpowder and it still ignited, that was proof that what you were purchasing was really alcohol. Now, I'm not talking about going to the store. I'm talking about, when people are selling barrels of alcohol to one another, so forth and so on. But if you wet the gunpowder and the gunpowder still ignited, that was proof that it really was alcohol. Whereas if that were a majority water solution and you were to wet the gunpowder, then of course the gunpowder is not going to ignite if it's not really of a sufficient alcohol content. Okay, now let's talk about how we measure blood alcohol levels in the body. There are a couple of ways that we refer to it. Okay, we might say BALs, BACs, BELs, BECs. Okay, those stand for blood alcohol level, blood alcohol concentration, blood ethanol level, blood ethanol content. Okay, all of those things mean the same thing. We're talking about how much alcohol is in the blood. Okay, and so if you go back to chapter 1, similar thing when we talk about how much alcohol is in the body when we're looking at doses, levels. And so typically when we're talking about blood alcohol levels, blood alcohol content, blood ethanol level, blood ethanol content, we're talking about, okay, how many milligrams per 100 milliliters of blood, okay, are there in the body? How many milligrams of alcohol per 100 milliliters of blood are in the body? Okay, and so that 100 milliliters of blood is also the equivalent to a deciliter. Okay, and so this is why you see the term milligram percent, because we're looking at how much is in that 100 milliliters. Okay, and so whenever we use the percentages as a portion of the whole or 100. Okay. Now, probably the largest difference you see in whether or not we use BAL, BAC, BEL, BEC is more often than not, if you're reading a published research article and animals have been used in the study, you're more likely to see blood ethanol level, blood ethanol content. You're more likely to have an investigator refer to the substance as ethanol in an animal study. You will still see the word alcohol appear, but you're more likely to see ethanol, that term, probably because it's being more specific about the chemical name rather than just alcohols in general. But you're more likely to see ethanol used in published articles about animal studies that involve ethanol. Okay, and so how we measure blood alcohol levels in the body, blood sample, of course, since we've just talked a lot about blood alcohol levels, blood alcohol concentration, etc., etc. The other way that you can measure the amount of alcohol in the body is through use of a breathalyzer. Okay, the breathalyzer is a reliable way to measure blood alcohol levels because we know that alcohol vaporizes at a known rate, and so the air that you exhale, the air that you expire is going to reliably reflect the amount of alcohol that's circulating in the bloodstream. Okay, and so breathalyzer tests are quite accurate, and so it would take someone tampering with a breathalyzer or somehow screwing up the calibration for one of these tests to be inaccurate. Okay, and there are only a very few examples of individuals that have been able to successfully challenge the breathalyzer test. And they're quite sensitive. So if you have individuals that have, for whatever reason, drank a large amount of cough medicine, or drank a large, for whatever reason, drink a large amount of mouthwash or substances that may contain alcohol, then those can be picked up sometimes by these breathalyzer tests. Those are very sensitive tests. Okay. Now, what we're seeing on this slide is a curve, okay, that's showing you what your blood alcohol levels would look like after a single drink. And when we say after a single drink, okay, we are talking about equivalencies across the different types of alcohol. So if you had a standard shot, or a glass of wine, or a beer, but you're adjusting for the different concentrations of alcohol in those various solutions. So you're not having a gigantic shot. You're having a, I'm not going to try to give you the size of the shot now, because I may give the wrong thing, but, we're not comparing apples and oranges, okay? But this is what what your blood alcohol levels would look like after a single drink. This, of course, would look very different if an individual continued drinking. Okay, so we're going to look here at oops, sorry about that. This portion of the curve, this is the absorption phase, okay? When the individual just starts drinking, and the alcohol is being absorbed into the bloodstream. The blood alcohol levels are rising, okay? Now, obviously, because the individual is drinking and it's being absorbed into the bloodstream, your textbook says that absorption is taking place more rapidly than excretion. In actuality, you have very little to no excretion that's happening during this absorption phase. Okay, and then B and C, here from our textbook, we're combining these two parts. As an individual continues drinking and all of the alcohol gets absorbed into the bloodstream, the absorption is going to taper, and here you'll see that the blood alcohol levels peak. Okay, and then what happens at excretion starts to lower the levels of alcohol. Okay, and then we enter this excretion phase, this side of the curve, okay, where excretion is going to start to kick in. Okay, here you have no absorption happening, because remember, the absorption has been completed in this area of the curve. But during this excretion phase, the alcohol is going to be eliminated from the body, okay, so it's going to be metabolized and excreted at a constant rate. Okay, now let's say this: when we talk about alcohol being metabolized and excreted at a constant rate, we say that alcohol is following what we call zero order kinetics. Okay, with zero order kinetics, alcohol is metabolized and excreted at a constant rate. Alcohol is the only drug that we'll talk about in this course that follows zero order kinetics. And actually, I'm not familiar with another that follows zero order kinetics, although there may be some drugs out there that I'm not familiar about. Most of the drugs that we talk about in the course will follow what we call first order kinetics, where the rate of metabolism and excretion is going to be proportional to the amount that you take. But not so with alcohol. Alcohol is metabolized and excreted at a constant rate. And so one of the things that's going to come out in this chapter when we talk about this zero order kinetics is that my constant rate is my constant rate. Your constant rate is your constant rate. The constant rate of a social drinker is going to be different from the constant rate of an alcoholic. So if you want to imagine individuals on a highway, okay, you could have 3 cars that are all going constant rates. Okay, so how do we go constant rate on the highway? Let's say we set our speed control, or cruise control, that's what you call it. We set our cruise control. You have one car that has the cruise control set at 75. You have another car that has the cruise control set at 65. And then you have a third car that has the cruise control set at 55. Those three cars are traveling constant rates, but their constant rates are different. That's what we mean with alcohol. That when an individual drinks, that alcohol is going to be metabolized and excreted at a constant rate. But how that individual metabolizes and excretes alcohol is going to be different from individual number 2 and individual number 4, and it's certainly going to be different from the alcoholic. Okay? So this is our curve that shows us what our blood alcohol levels look like after a single drink. Okay, and I'll stop here for this section. Give you time to think about this portion.